Thank you. So I think a lot of people have heard of Ripple, but a lot of you don't really know what the difference is with Ripple and other drug delivery technologies. So my goal today is really to explain the Ripple difference. And the first and most profound difference is we've discovered an ability to deliver drugs over a period of time without polymers or excipients. And this has a number of benefits. From a PK point of view, smaller implants, higher drug loading, zero-to-release kinetics that are very tunable for dose and duration. From a clinical point of view, the goal is to be in the therapeutic range for longer without the burst release, which allows for both a reduction in treatment burden and improved long-term outcomes. And because these are NCEs, we can get composition of matter patents. And despite the fact there's no polymers, we can use standard polymer processing techniques, which are very scalable. The other difference is the breadth of the technology. We started with dexamethasone and other steroids in 2017, and you can see we've built that platform significantly. We think over time we can even start working in new drug classes, including things like aptamers and peptides. We've also been able to demonstrate the translation from the benchtop to the bedside. So this is an example of IB814, which is our intravitreal dexamethasone implant for DME and RVO. You see on the top left there the PK study relative to Ozurdex, typical burst release Ozurdex, and our nice, steady zero-release kinetics that translates really nicely to the PD on the bottom left. And the key thing is we've been able to translate this to the clinic. So this is just two examples, two patients, one in DME and one in RVO, with very long treatment duration. And you can see the nice reduction in CST in one case for 9 months and the other case for almost 10 months there. We have an experienced team that's complemented with a with a board of directors that has deep ophthalmology experience, and both scientific and medical advisors that support us. So because of the breadth of this technology, we've been able to address two different pathways from a commercial point of view. The first is developing proprietary products, which we can either develop then on our own or partner to companies depending upon the economics of the deal. And we announced a deal last year with AbbVie where they licensed our bimatoprost intracameral implant. On the other side are funded partner programs, and we announced a deal last year with Glaukos. So the AbbVie deal was really transformative for Ripple, not just because of the economics, but also because of the validation of our technology platform by one of the biggest players in the space. Funded partner programs are a little different. This is where a partner comes to us with their own molecule and is interested to see if our technology can work with theirs to develop a sustained release approach. So they fund the development program and then have an option at the end of the development to a pre-negotiated licensing agreement. So it's a fully funded program, and if these products are successful, significant economic upside. So from a proprietary product point of view, we've really focused in two areas. One is a Tanaprost intracameral implant with a 12 to 18 month duration, and then two different approaches for dry AMD, and in the interest of time, I'm just going to give you a little bit of data on those two. So the first is targeting a complement factor inhibitor, and we think we can take advantage of the established regulatory and commercial pathways, and the fact that we're at the target upstream from C3 and C5, and we don't have any of the polymer requirements that those do, we think we can have an improved safety and efficacy approach. It's early days here, but we've just completed tolerability studies, and these are looking good. The second is more unique, and that is using intravitreal statins to address the multifactorial issues with dry AMD. We think the literature is now supporting this approach, and that intravitreal statins are really the way to unlock the capabilities of this drug. Good progress so far. We've got 4-month ocular tolerability. We've started our PD studies, and those will be available in the next few months. Our goal here is to get to the clinic as soon as possible, and that's really the focus of our latest financing. And the last thing about difference is really capital efficiency and economic potential. So we've secured over $65 million in funding, but only $15 million in equity, the rest being from partner programs as well as non-dilutive funding. And as I mentioned, the partner programs are fully funded, so these are all upside. And we're focused on proprietary products in very significant market opportunities. So the Ripple difference is really about a scientific discovery that is unique in terms of being able to create these sustained release implants without polymers or excipients, a versatile program that we've proven clinical translation, experienced team, and really this balance of proprietary and partner programs allows us to have a very diverse and robust commercial opportunity. Thank you for your time.
Thank you. Any questions? I think it's great technology. I think the fact that you can bypass the polymer and get zero order kinetics is great, especially in the light of PEG and other things maybe causing inflammation with some of our newer agents. How are you able to package like peptides? I mean, how can you do that? How can you squish the peptides together and
that's
not
early.
have them
So
bind to themselves?
Yeah, we're not doing that right now.
Okay.
We think that that's on the horizon, but right now we're focused primarily on small molecules. But if you look at the difference between a steroid and prostaglandins, you have prostaglandins, which are oils. So the versatility of the system, I think, is evident in the differences in the molecules we've already worked on.
Yeah, what steady dose of statin are you trying to achieve in the eye?
Well, we're not talking specifically about the dose right now, but we think that the intravitreal approach is the key to getting enough drug in the back. There's been studies, I think this Geimer paper back in 2013, which looked at the impact of statins on the target tissues, but the problem is taking oral statins, you just need too much to get to the target tissues. So we think the intravitreal approach is the right one.
You did share the dose of dexamethasone in the whatever, 85 nano or micromolar, whatever it was. Anyway, how does that compare to like Iluvien in efficacy? Because Ozurdex has a burst and it's helpful, and Iluvien doesn't have the burst, and you don't really see what's going on. You certainly see what's going on with your drug because the thickness is really remarkably better.
Right.
So I assume the efficacy of your dexamethasone is higher than the comparable efficacy of Iluvien. Or do you know that?
So we've designed it to be a 6-month release with the 30-gauge needle because that's what the retinal specialist said that they wanted. We also did studies with 24-gauge, which lasts 24 months. So it's very versatile in terms of the ability to moderate that. But the goal here was to be able to deliver 6-month therapy with a 30-gauge needle.
Okay. Thank you very much.
Thank you.